In this TA Tech Tip, we're going to show you how to set up the photocalorimeter accessory for use with the TA Instruments Q2000 DSC. The photocalorimeter accessory, or PCA, allows samples which have been placed inside the DSC to be irradiated with ultraviolet or visible light. The light source contains a high pressure mercury lamp delivering the light. Typical irradiance levels for DSC experiments are 20 to 100 milliwatts per centimetre squared. A range of filters are available to control the spectral range of the light used for the test. A standard filter is present in the light source. Additional filters are available through the use of an external filter adapter. These provide spectral ranges of 320 to 390 nanometers, 365 nanometers and 400 to 500 nanometers. Visible long-pass filters with cut-offs at 390 and 490 nanometers are also available. Two types of light guide are available. Quartz to operate from -50 to 250 degrees Celsius and liquid to operate from -50 to 80 degrees Celsius. Filter holders at the end of the light guides are used to hold cut-off filters and neutral density filters. The light guide adapter holds the light guides as a single unit. The Q2000 design technology allows the heat flow of the reference and sample platforms to be independently measured. The light guide adapter base is fitted to the top of the DSC cell. This provides repeatable location of the light guides in the adapter and can be left in place when running a standard DSC test on a sample which has just been studied using the PCA. In the controller software, make sure that the instrument preferences are set for the PCA. Setting the PCA option disables the auto sampler and auto lid, as these are not used when operating the PCA. Unselecting the PCA option will not re-enable these. This allows standard DSC tests to be run using the manual lids provided without removing the light guide adapter base. The auto sampler and auto lids are re-enabled using the options in the control menu. Ideally, you should also have the sample and reference intensities set as two signals available on the touchscreen. This helps when setting the irradiance levels. Connect the RS232 cable to P3 at the back of the light source and COM2 at the back of the DSC. This provides the shutter control from within the experimental method. Open the auto lid and place the light guide base and adapter on the DSC cell. Make sure that the light guide positions are aligned with the sensor platforms and fix the base in place by tightening the 4 outer screws. The 4 inner screws can be used for small adjustments of the alignment. The dual light guide is fitted into the light source and automatically locks into place. The filter holders are fitted to the DSC ends of the light guides. Both have small containers into which neutral density and/or long pass filters are placed. Use matching filters for both sample and reference guides. The light guides can now be fastened into the adapter. This allows them to be fitted or removed from the adapter base as a single unit. The light source can now be turned on and allowed to warm up. The LED lamp will stop flashing when it is ready for operation. The PCA can operate in two modes. The default setting is a relative mode where the intensity from the light source is set as a percentage of the total output. An absolute intensity may also be set. This requires calibration of the intensity with a radiometer. As part of the setup, we've inserted the required filters. We can now balance the intensity on the sample and reference platforms. There are vertical positioning screws in the light guide adapter. Make sure that these are set to the lowest point. Using an empty cell, fit the PCA silver lids and quartz window and the light guides into the adapter base. On the light source, press the mode button until the level option is active. Then, either from the control software or from the front panel of the light source, open the shutter. Remember that we are dealing with UV light, so suitable protection should be worn. This should include covering arms, wearing latex gloves and wearing UV eye protection. Select the signal view on the touchscreen so that you can see the reference and sample intensity signals. Using the up and down arrows on the light source, set the lowest of these signals to the intensity you require. Then, adjust the vertical positioning screw on the side with the highest intensity to raise the light guide and lower the intensity so that it is within plus or minus 2% of the required value. In this TA Tech Tip, we have talked about the installation and initial setup of the photocalorimeter accessory for the Q2000 DSC. Please stay tuned for 2 associated tech tips, one talking about the experimental setup and approaches, and the other talking about data analysis. Thank you for your interest.